inspiring the next generation of leaders like yourselves. So before hearing from our incredible speakers, I'd like to thank today's partners. In particular, I'd like to thank our presenting sponsor, the Krembil Foundation. The Gairdner Foundation is also grateful for the support of the Government of Canada, CIHR, the Government of Ontario, Trillium Therapeutics, the Lunenfeld Tenenbaum Research Institute, Sick Kids Research Institute, and the Sunnybrook Research Institute for allowing us to put on important events like today. If we could have a quick round of applause for our sponsors, please. So thank you for joining us, and now please join me in welcoming our chair for the day, the Scientific Director of the Krembil Foundation, Kate Williams.
Good morning, everyone, and thank you, Summer, for that introduction. I'm thrilled to be here today on behalf of the Krembil Foundation, and we're very excited to be partnering with Gairdner to bring you today's Research Symposium in Optogenetics. For those of you who aren't familiar with the Krembil Foundation, I'll give you a very brief overview of what we do. Since 2001, the Krembil Foundation's mandate has been to help people by supporting medical research and education. Our primary focus is funding cutting-edge, novel science, and neuroscience has been a priority since the beginning. We focus our support on basic science because we believe that a fundamental understanding of the brain is critical for understanding brain disease and developing treatments and therapeutics. We're interested in new and exciting research that has the potential to produce life-altering results, much like the work of the incredible researchers and scientists that you'll hear from today. Today's speakers are some of the world's most distinguished sources on optogenetics. During the symposium, they will explore the advances and uses of this technology in neuroscience and their own specific research questions. I have no doubt these talks will both enlighten and inspire all of us. Our first speaker today is Dr. Leslie Boshall. Dr. Boshall is a molecular neurobiologist who studies how behaviors emerge from the integration of sensory info with internal physiological states. She is the Robin Chambers Newstein Professor and head of the Laboratory of Neurogenetics and Behavior at Rockefeller University, as well as a Howard Hughes Medical Institute investigator and director of the Kavli Neural Systems Institute. She received an A.B. in biochemistry from Columbia University and a Ph.D. from the Rockefeller University. Following her postdoctoral work at Columbia University, she joined the Rockefeller faculty in 2000. She is known for her work on the genetic basis of chemosensory behavior in both insects and humans. Much of her current work is aimed at understanding the molecular neurobiology of host-seeking and blood-feeding in mosquitoes that spread infectious disease in humans. Please join me in welcoming Dr. Boshall.
Good morning. I'm really thrilled to be here to celebrate with you the Gairdner Foundation Laureates of the Year, and also to celebrate optogenetics. The specific technique we'll be discussing today really has been transformative for biology, allowing people asking all sorts of different questions to make big advances in biology. So my research program is somewhere in between the Global Health Prize and the Neuroscience Prize because we study this animal, the mosquito, in our lab, Aedes aegypti, that spreads viral diseases to hundreds of millions of people every year. I'll divide my talk into two parts. So first, I'll tell you about how we use optogenetics to activate sensory pathways to understand how multiple different sensory cues activate behavior. And in the second part of the talk, I will discuss a new taste quality, how mosquitoes actually get really excited by the taste of ATP. So first, something about mosquitoes. So these are fascinating insects, but they're also deadly insects. So a lot of people don't realize that in North America, until early in the 20th century and even mid-20th century, there were still vector-borne diseases. So you can see here in the southern states of the United States, there was still active malaria transmission. And in fact, malaria was not eradicated from the USA until 1960. A lot of you didn't know this. And Europe took even longer to eradicate malaria, 1975. So sort of 3/4 through the 20th century, we still had active mosquito-borne disease in Northern Hemisphere countries. And what's remarkable is that these mosquito-borne diseases can come back because the mosquitoes are always there waiting to spread new diseases. You just need to have infected people coming in, causing local infection of the mosquito population, and they will infect local people. And this can often be a result of political instability, and so you can see there was a huge rise of malaria cases coincident with the first Gulf War and the dissolution of the Soviet Union, when there's mass migration of people and political instability. So at the peak, almost 100,000 malaria cases, and this was sort of at the transition to our century. So of course malaria, although it has been eradicated now from Europe and from the United States, remains one of the biggest global killers, and killing in 2016 almost 500,000 people, mostly children under the age of 5 in sub-Saharan Africa. The fact that this number is half what it used to be is thanks almost entirely to the Bill and Melinda Gates Foundation, which have pushed hundreds of millions of dollars into the problem of trying to eradicate malaria. So thanks to Bill and Melinda Gates. So just as you can have malaria reoccur with political instability, the mosquito we study, Aedes aegypti, is able to pick up arboviruses wherever humans bring them. And so this is a picture of the worldwide distribution of Aedes aegypti. I think Canada is spared, but certainly throughout the southern US and the rest of the tropics, you find them. So what kind of diseases does Aedes aegypti spread? Dengue, an emerging tropical disease that infects almost 400 million people a year worldwide and causes terrible symptoms. Yellow fever, for which we have an excellent vaccine, but which was a big killer before there was a vaccine in the mid-20th century. And then two viruses that until recently no one had heard of. So chikungunya emerged in the mid-20th century in Kenya and was sickening people throughout Africa and Asia, but had never been seen in the Western Hemisphere until 2013, where people started getting sick around the St. Martin Airport in the Caribbean. Samples were sent to the CDC, and it was discovered it was chikungunya, a virus, again, that had never been seen before, presumably brought in by a sick person who landed at the St. Martin Airport. Wanted to have a great vacation in the Caribbean. 9 months later, 1 million people were infected with chikungunya throughout the Western Hemisphere. It's not that the mosquitoes are flying from Florida down to Brazil, it's that people are flying from Florida to Brazil. The local mosquitoes are waiting to infect local people. And then quite notoriously, Zika, which emerged in 1947 in Uganda, a virus that made its way around the world via Thailand, French Polynesia, landed in Brazil in 2014. What started out as a pretty mild flu-causing virus, of course, is now strongly implicated in microcephaly, so pregnant women have a hugely elevated risk of giving birth to microcephalic babies, and Zika also, in the people who are infected, vastly increases the chance that they'll get an autoimmune disorder called Guillain-Barré. So just to show that we in the North are not safe from mosquito-borne diseases. And my whole research program, which bridges public health and neuroscience, aims to understand this biology. So this is a female Aedes aegypti. She's feeling really good because she has found a human. She has inserted this organ, which I'll talk about later, called a stylet. She's unsheathed it, and she has injected the stylet into the skin, where she obviously has found blood. She has doubled her body weight with this blood. And so we really want to understand, how are these females finding humans? How are they detecting the blood underneath the skin? And what can we do to stop them? So a really important thing that you can take away from here is that males don't bite. So the females need the blood to produce eggs. Basic fact of biology, males don't produce eggs. And so the female has really a nervous system that's wired to hunt humans. Males are rarely found on humans unless they are trying to pester the females to mate with them. The males also lack that syringe, that sharp syringe that the females use to puncture the skin. So really fascinating sexual dimorphism, both anatomically and neurobiologically, where the will to hunt and the will to bite is absent in males, something that we're really fascinated by. So an important aspect of these mosquitoes, both for the malaria mosquito and the mosquito we study, is that they specialize on humans. So in this really cool study from Laura Harrington, she and her student went around this village in Thailand and collected mosquitoes. They also counted the number of humans wild animals and domestic animals, and then immunotyped the mosquitoes and asked what kind of blood do they have in their midgut. And you can see there were a lot of dogs, goats, and pigs in this village. The mosquitoes had free access to them, but the vast majority of them fed on humans. And so this is why they're so effective at spreading disease among humans. They ignore other animals, passing these pathogens between people. So this is my former postdoc, Lindy McBride, where we offer these mosquitoes a choice of the smell of Lindy or the smell of Rosie, the guinea pig. And the animals, you can do this with malaria mosquitoes or arbovirus mosquitoes. 90% of the time, they find Lindy far more attractive than the guinea pig. And so why is that? So whether or not you took a shower, whether or not you wear perfume or deodorant, we all have a unique human scent. And if you look at the chemistry of human skin, so this is just a gas chromatogram where you have different peaks that elute at different times. At the top is the guinea pig, at the bottom is the human. You can see some similarities, so some compounds are in common, but there's a vast number of differences. So if you have a guinea pig, you smell your guinea pig, obviously smells different than you. The mosquitoes are very sensitive to these differences, and that is a feature by which they make decisions for whom to bite. And so this is the kind of stuff that's in your skin. Again, whether or not you shower, the microbiome on your skin is busily digesting the sebum and other products on your skin to produce this perfume that's highly attractive to mosquitoes. So this is what the behavior looks like. That my lab is really driven by the genetics of behavior. So here's a small patch of human skin, human PhD student skin, fully vetted by the ethicists in our lab and in our institution, and I do this also. So this is sped up, and you can see that although the mosquitoes are flying in a really large cage, they have really rapidly found this really tiny patch of exposed skin. This person is breathing into the cage, so carbon dioxide has activated them. There's heat coming off the skin, and there's some of those molecules coming off the skin. So, and you can see them filling up with blood that will then nourish the next generation of about 100 progeny, 50 of whom will be female, who will go on to bite additional people. So we really want to understand how can they get to the skin so quickly. So this is an optogenetics symposium. I felt under pressure to give you some optogenetics data. So my postdoc Trevor Sorrells has been working hard the last few months just for this symposium to show that we can use optogenetics to study multimodal integration. How do animals put these different cues together? So this is you, or a cartoon man, with a mosquito trying to evaluate, is this a guinea pig or a human? Obviously humans look different than animals. We also, as I said, smell delicious, so there's all of these molecules that are a signature of humanness that they love. If we are alive, we exhale carbon dioxide. Laundry smells good, but it does not produce carbon dioxide, so it's not attractive. And we're also warm-blooded. And so all of these cues the female puts together to have an honest signal of, is this a human? And so there's lots of behavioral evidence that these animals integrate multiple different sensory cues, including all the 5 here. So the question that we've been asking is, do they use just one cue? Do they require 2 cues, or do they need all of them? And this makes a lot of sense. Again, heat, there's lots of sources of heat in this room. If you were a mosquito that flew to the heat, you would just find a radiator and not a human. If you only paid attention to carbon dioxide and flew to a can of Diet Coke, you would not find a human, you would only find carbon dioxide. So it makes a great deal of sense for them to pay attention to multiple sensory cues. So early in my research program, we used zinc finger nucleases, a technology that has been forgotten because CRISPR-Cas9 is now, of course, the way to do genetic engineering in biological and medical sciences. We use these techniques to eliminate the ability of mosquitoes to smell carbon dioxide so we could test what are the consequences on their ability to hunt humans. And so this is just a picture of what we did. Again, the theme here would be that you devise either a protein or an RNA to recognize a specific gene sequence. The protein machine, usually Cas9, will cut the chromosome, either generating mutations or allowing you to integrate a marker so you could follow your mutants. And so one of the first genes that we knocked out was a gene called gustatory receptor 3. These animals have this unique property, insects have this unique property of being incredibly sensitive to carbon dioxide, and they do it with this multi-subunit machine of gustatory receptors 1, 2, and 3. And we had some evidence that gustatory receptor 3 was one of the most important ones. So Connor McMenamin knocked out this gene so we could ask what happens if you have a mosquito that can't sense carbon dioxide. So here's a picture of the beautiful offspring. The mutant is on the left, the wild type is on the right, and you can tell it's a mutant because it's glowing bright blue. So this is the magic of genetic engineering, where you can knock out a gene and also mark the mutant. So what is the phenotype of these animals? CO2 really potently activates mosquitoes, and so Ben Matthews in the lab set up a multi-insect tracking paradigm where you put a lot of females into a cage and you can follow their flight paths, and then you can puff in carbon dioxide and see how the flight path changes. So you can see here in this 6-minute period they're flying around, you add CO2 and they really go berserk, they really get excited. If you take away one copy of gustatory receptor 3, they're still very excited by carbon dioxide, also plotted here. So what happens if you take out one of the 14,000 genes of the mosquito? They become completely insensitive to carbon dioxide, which is remarkable. So again, 6 minutes flying around without CO2. With CO2, they don't detect it at all. They just continue flying around. What's really cool is that the sensation of carbon dioxide influences the sensation of other cues. So this is a minimalized assay asking, do mosquitoes respond to heat? And so I'll draw your attention to this little plate here, a Peltier that we can heat up to human body temperature. And so in this video, you see mosquitoes just hanging out with ambient carbon dioxide. We'll shortly heat up that plate and then put CO2 into the cage. So you can see them now with your own eyes going completely berserk. And you can see that they have detected there's something interesting here. So they start to accumulate on this plate, because again, they have now two cues, heat and carbon dioxide, which in their little tiny female mosquito brains means human, 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 human. The box is quite big relative to the scale of the mosquitoes. If you look at what they're doing, you can see that they're probing and biting. And Ramon has a way to track these animals on the side of the cage. So if we look at the percent of animals that go over to that Peltier, if you turn up the heat and you add carbon dioxide, they love it. So half of them go onto that area. They're trying to probe and bite, frantically trying to find the human that's fictive, that's not there. And then immediately after you turn the heat off, they lose interest. This critically depends on both of those cues. So if you supply the heat, it's the same heat, the heat is still there, but you have not activated them, the mosquitoes have no interest. And of course, if you just put in CO2, there's no heat, they don't go. So this is a really clear demonstration that these two sensory cues have to be integrated for them to elicit this behavior to try to bite. And very gratifyingly, we have, as I showed you, we've made mutants that can sense CO2, and these animals are, whatever combination of genetic mutants we make, none of them are able to seek heat. So they're blind to one stimulus, which makes them blind to a second stimulus, like a really formal proof that multimodal integration is happening. And this gets even more profound. We then ask them to, we offer them a blood meal, and without carbon dioxide, the controls, 100% of them will blood feed, and the mutants have a really hard time going to that same membrane feeder to find a blood meal. So the single sensory cue in these laboratory experiments contributes a great deal to the behavior. So, okay, here's the optogenetics part. If carbon dioxide detection is, I showed you it's necessary, you knock out the CO2 receptor, they no longer respond to CO2, they no longer respond to heat, they have great difficulties feeding on blood. So a gain-of-function experiment is enabled by optogenetics, this amazing technique taking basic research, identifying a protein that you could transfer to invertebrate neurons and get them to be activated. We have just very recently, thanks to my postdoc Trevor Sorrells and a technician Anjali Pandey, been able to make optogenetics work in the mosquito. And so the idea is, if you take neurons that are causal in behavior and you either activate them or inhibit them, you should be able to figure out if that has the power to drive or inhibit behavior. And so we've been using a variant of channel rhodopsin called CS crimson that works very well in insects to try to figure out which of these different behavioral states of resting you saw, searching you saw, feeding you saw, is impacted by activating the CO2 circuit. And so we hypothesized that activating just those neurons should cause them to become activated. And so that's absolutely true. So on the left side are the crimson animals. The LED has just gone on. On the right side, it's not a still video. The animals just don't care. So you saw, some number of these animals. They were just hanging out, grooming, minding their own business. The light was turned on. See that again because it's so cool. So they're just hanging out, grooming. Shortly the light will go on. And they increase their movement and go berserk. And so that's what this looks like. So over 50 different mosquitoes, you see that the average activity, even with a 1-second stimulus, causes a huge increase in the base velocity of these animals, and it lasts for quite some time after the light turns off. And it does scale with the amount of light that we give them, so that suggests that, again, neural activity in these CO2 GR3 expressing neurons will scale to locomotor behavior. Very gratifying. So Anjali and Trevor designed this really clever device that he calls an optomembrane feeder, where you have a cylinder of light, so let there be light. It's a whole cylinder of LEDs, and then a bit of blood is brought into position, which is indicated here. So the mosquitoes are in here. They're offered some warm blood, no carbon dioxide, no human body odor, just light. So if we just activate these neurons with light, and we offer them this delicious meal, with the only cue being the heat cue, will they blood feed? And so that's shown here. So on these two left panels are the controls, and the right side is the animals that have optogenetically activated carbon dioxide receptor. Throughout this experiment, the lights are flashing on and off because it's a long experiment. You see they're kind of hanging out. This circle is where the blood is. And so you see that the animals on the right have now found the blood, and in this time lapse you can see that they are filling up on blood. So this works. We have been able to activate a few dozen cells using an optogenetic variant of channel rhodopsin, and we have been able to trigger blood feeding. These animals over here, the control animals, are sitting over the blood, but they are not initiating blood feeding. I thought this was one of the coolest things I've seen in my lab in recent years. So I think the next step is to figure out how's the brain dealing with this information. You activate sensory neurons. How is the brain taking this information into context? CO2 causes activation. How do these animals figure out that it's a human and not a guinea pig, that it's a live human? And so Meg Younger has been working to get into the brain of the mosquito. You guys can go to mosquitobrains.org and see the beautiful brain. So I'd say the mosquito brain is not that different from the vertebrates that you'll see in the beautiful subsequent talks. So we have an olfactory center in blue, we have a taste center here in cyan, and then visual centers here, and then this is the fly hippocampus right here. And here's the fly entorhinal cortex. So all the great stuff you'll hear later, flies have it too. So what Meg has done is ask, what does it look like when the brain detects carbon dioxide? So we've been able to, in the last year, make enormous strides in taking the tools that people have used in the mouse, the fly, and in C. elegans to do mechanistic molecular neuroscience in mosquitoes. So here's an animal where we have taken those GR3 neurons and put in them a genetically encoded calcium sensor, GCAM6S, pioneered by Lauren Luger and colleagues at Janelia. And so here you see all of the neurons coming from the antenna, projecting to this one area of the antennal lobe, which processes scent. And so the hypothesis would be if those neurons are sensing carbon dioxide, which we have genetic proof for, if you puff carbon dioxide, you should see activation of that part of the brain. And that part of the brain is here. So this is a huge glomerulus, so a huge part of the mosquito brain is set aside to detect carbon dioxide, an important stimulus for these animals. And so this works really beautifully. So when Meg puffs on carbon dioxide, you can see that there's a really large increase in the activity of calcium in these cells. And we're so excited we can do all this cool stuff, like knock transcription factors in, make all these different effectors. And so you see this beautiful dose-dependent increase with increasing concentrations of carbon dioxide. So we're really poised at this moment in the history of mosquito neuroscience to really dig in and figure out, as CO2 is coming into the MD1 glomerulus here, how is the odor of humans coming into this structure? And then how is that integrated? How's that different in males? So males have GR3 expressing neurons, but they don't respond to the integration of heat and carbon dioxide. So is it that in development the male is wired differently, or is the wiring the same in males and females, but there's some kind of global inhibition that tells the male, don't behave like a female, you don't need to hunt humans, just ignore it. All this sensory stuff may be coming in, but perhaps he's ignoring it. And so with the tools that we've built in the last few years, I think we're really poised to make some advances here. So the second part here is this discovery that my student Veronica Jové has made about what is it that these animals are tasting when they put their syringe underneath the skin. So a lot of this work is with this membrane feeder, not the skin of people in my lab. So if you heat up a bit of sheep blood here, again, puff in carbon dioxide. Parafilm kind of mimics the puncturing of the skin, and also there's a dirty sock wrapped around this, so great stuff. The mosquito loves this, and you'll see her midgut swelling with this. Doesn't look like a human host, but it sure smells like a human host. So we've been interested in what's going on underneath the surface here. What is this female doing? It isn't like, humans are not like this. We're not a piece of parafilm stretched over human blood, right? There's a lot of anatomy, a lot of cellular anatomy when you puncture the skin and try to find a capillary. And so a group at the Pasteur made this amazing video of the stylet searching around underneath the skin of a mouse. And so the red bits you see are blood capillaries. This brown thing is the syringe. This is the stylet. And it's just like when you go to the doctor's office and they want to draw your blood, where the phlebotomist inserts a syringe. And so we're really fascinated by this structure. So how did this stylet find that capillary? It doesn't have eyes on it, right? The mosquito's on the surface. Is it sensing, is there increased warmth coming from those capillaries? Is ATP being produced? This is an intensely inflammatory response. You know, if you puncture your skin with a needle, you know, it hurts, and there's an inflammatory response which releases ATP and other immunogenic inflammatory molecules. When these animals are squishing blood cells, they should be releasing protein and a lot of ATP, and perhaps it's also sensing flow. There has to be some way for it to turn on the pump. And so what Veronica has been studying is what is causing that stylet to do its thing. And so again, I just want to point out that only females blood feed. So the one thing that you leave here is like, please don't tell me at the break that males blood feed. So females only feed on blood when they need eggs, and both males and females feed on floral nectar, just as an energy source. And so this is the stylet. So this is the stylet. The labellum here, the tongue has been retracted, and the stylet's underneath the skin. And so what Veronica's been doing is trying to figure out what is unique in gene expression and neural function of this little tiny sensory structure that separates it from the labium, which is, again, the insect equivalent of the tongue, that causes these animals to sense something under the skin and fill up. So work from the 1960s by Rachel Galloon showed that ATP, the universal currency of energy, the universal currency of life, ATP can trigger blood feeding. So this was a really fundamental insight. So here's just a picture of a female mosquito. We've given her carbon dioxide, we've given her odor, we've heated up a bit of saline and put ATP in it, and she drinks it hungrily. So there's something about this really, of course, blood has hundreds, thousands of components in it, vitamins, proteins, lipids, and ATP, but all the mosquito appears to need is just a taste of ATP. Insects do not have P2X receptors. that would allow them to sense ATP. So this is a complete mystery we wanted to solve. How are these animals detecting ATP? And so here's just the data showing that if you look at the percent of females that engorge on blood, they will engorge on blood. Even though they like sucrose, sucrose is not a signal that causes them to engorge. It's not a signal of a blood meal. The saline does not excite them, but you put 1 millimolar ATP in there and it's indistinguishable from blood. And so you can see here at the left, some poor female who's been tricked into drinking basically water, and which will not produce eggs. But she was excited by it. So Veronica asked this question, what is it about the taste of ATP? Does it require, is it some local event where you need to hydrolyze ATP? Is it acting in some sense as an energy source or some sort of a pump? So a really great test of that is to put non-hydrolyzable analogs in. And remarkably, each of these 3 non-hydrolyzable analogs is as good at ATP in triggering engorgement. And they like it just as much, so if you weigh them before and after, they more than double their body weight with ATP and these analogs. So whatever we're looking for does not require ATP hydrolysis, but must be some receptor mechanism that can detect these small molecules. This is exquisitely dose sensitive, and so if you reduce the dose of ATP, you start seeing significant triggering of engorgement as low as 1 micromolar. What's cool about this, it's binary. It isn't that at 1 micromolar, these animals take 1/20th the concentration. It just decreases the chance that they'll engorge. So ATP, these animals seem to be poised at this moment of should I turn on the pump, should I not turn on the pump? And the stronger the signal of ATP is, the more of them will flip over to eating. So if we look inside the stylet, you can see that, so it's tiny, right? This is the diameter of a mammalian neuron. So this is an incredibly tiny structure. Insect neurons are much smaller. These creatures have mechanosensory neurons here. So this is a single mechanosensory neuron here that probably is sensing that movement as it's going around. And then a number of chemosensory receptors. These are our candidates for sensing either protein, ATP, or other compounds. And what's incredibly cool is that the sexual dimorphism behaviorally is reflected at the sensory level. So on the right is a male stylet, and so you see that it's much less sharp. It doesn't have this tight bevel, and most importantly, has very few neurons. It does have this mechanosensory neuron here, but it lacks the chemosensory neurons that go into the tip. And so the idea is that we think that ATP detection must be female stylet specific. And so here's just phalloidin staining showing again that the sensory dendrites that are out there in the periphery where they would be sensing all of these things. The males lack the sensory dendrites and they lack many of the cells. So what Veronica did, which is another transformative technique in biology, is to take these tiny structures and profile RNA expression. So if it's female specific and it's stylet specific, we should find genes that are only expressed in female stylet, not male stylet, and also not in the tongue, the labellum. So if this is a specific taste quality, only in the stylet, only genes expressed here should exist and should be candidate ATP receptors. And so months and months of dissecting this 20 micron diameter little structure yielded these beautiful results that she finds 51 genes that are highly enriched in this syringe. We figured that an ATP receptor should be a membrane protein. We set the filter at it has to have more than 2 transmembrane domains. We find 12 of those, and 4 of them are exquisitely stylet specific. And so here's what they look like. So these are just transcripts per million reads of these 2 genes of unknown function. So you can see this gene is highly expressed in this female stylet, essentially undetectable in the female labium, tongue, and entirely undetected in the male stylet. This is the dream of a molecular biologist where your hypothesis actually works out, and to find multiple genes is even better. So we have this embarrassment of riches that Veronica is working with. These 2 genes, IR7A and IR7F, we know are chemosensory receptors. They are members of a huge expansion of ionotropic glutamate receptors. Glutamate receptors, of course, in the nervous system, invertebrates are the major mechanism of fast neuronal transmission. They sense AMPA kainate glutamate. In insects, these have been repurposed to be chemosensory receptors. They no longer detect neurotransmitters. They seem to detect sensory stimuli. And so we were really intrigued that these may be candidates for ATP receptors. And so what Veronica has gone on to do is to try to get into these neurons to figure out, do they respond to ATP? So she and other people in the lab have been working together to create genetic tools. And so we have made animals that, that express a co-receptor of the ionotropic receptor family that will partner with IR7F, IR7A, to generate some kind of a sensory receptor. And you can see that neurons expressing this co-receptor are abundant in the female and project their sensory dendrites up into the tip, and that males totally lack these. I feel sorry for the males sometimes. And so the question is, do these cells respond to ATP? So here's an animal where Veronica's taken another co-receptor in the ionotropic receptor family and expressed GCAMP6S, which is a genetically encoded calcium sensor. And I'll have you pay attention to these cells here. So this is the baseline signal. She's going to put in, she's going to transition from flowing saline to ATP. And there it is, this like amazing results, years and years of work. You see this single cell strongly lighting up to ATP, and we see no response when the cell transitions from water to saline. And so these are the cells that we've been imaging from, these beautiful sensory dendrites that we think are sensing ATP. Here's another example where she's been able to image all of these different cells. Right, one hypothesis might be that they all respond to ATP. The other hypothesis is that they're sort of dedicated to different taste qualities, and it's really the latter that's the case. So again, here we have saline going on, and then we have ATP in saline. So you see, again, cells 6 and 8. It starts to look like the kind of sensory code that is universal in sensory systems, where you dedicate different sensory neurons to different taste qualities. And so we see this huge selective increase when you switch to ATP in these female stylids. And just to convince you, here's another example. So water to saline. We do find some cells that are activated a little bit by saline, which will make sense. Saline is another biological signal, but the ATP response is very strong. And so it is selective to ATP, and also is activated by these non-hydrolyzable analogs. So here you see this response to AMP, PNP, and there is no response to ADP. So it perfectly matches the behavioral discoveries made by Rachel Galloon in the 1960s, that there's something in these neurons that is responding selectively to ATP or its non-hydrolyzable analogs and not to ADP, again, because the currency of life is ATP. So, and so here's just the data showing that for all of these different, for this one analog, that the response to ATP and the response to the non-hydrolyzable analog is equivalent, and this analog that ADP does not cause the response. We also don't see a response in males, not that surprising because they lack chemosensory receptors. So when we switch from saline to ATP, there's no response in that kind of sensory neuron. Okay. And just in a control experiment, we can show that in the female, there's also no response to anything except depolarizing stimulus of ATP. So where we are with this is we have 4 candidates that are highly enriched in the female stylet. They encode membrane proteins. 2 of them are members of this large family of ionotropic chemosensory receptors. We have this perfect correlation between the behavior, where we have neurons activated by ATP that also activate behavior, neurons activated by non-hydrolyzable analogs that activate behavior, and then ADP that does not. And so we're really excited about trying to figure out which or one or more of these genes is involved in ATP. That would be the first instance of protein in insects that senses extracellular ATP. It also gives us access to the circuits that trigger responses to ATP. And so we know that these neurons project to this taste area of the brain. I showed you here, this is the CO2 responsive part of the brain. And so this part of the brain seems to be uniquely specialized to sense information from the stylet. And so we're super interested in trying to understand how activation of these neurons leads to this taste behavior. So Veronica Jovet carried out the work on the taste of blood, graduate student in my lab, and Trevor Sorrells has been doing this beautiful work getting optogenetics to work with additional collaborators in my lab and the generous funders who support our work. Thank you very much. Other questions? No? Okay.
Thank you, Dr. Balasal. And in the interest of time, we'll be saving questions for after the symposium. There'll be a chance to interact with all the speakers. Thank you for a fascinating talk, and I congratulate your brave grad students for donating their arms and skin. Our next speaker is Dr. Hailan Hu. Dr. Hu is professor and senior investigator in the Center of Neuroscience at the School of Medicine at Zhejiang University. She received a BS in biochemistry from Beijing University and a PhD in neuroscience from UC Berkeley. Before joining Zhejiang University, she was a principal investigator at the Institute of Neuroscience Chinese Academy of Sciences. Her laboratory seeks to understand how emotional and social behaviors are encoded and regulated in the brain, with a main focus on the neural circuitry underlying depression and social dominance. Her work has led to the identification of several molecular targets for developing new antidepressant drugs. Please join me in welcoming Dr. Hu.
First, thanks, Summer, for the introduction. I also want to thank Sheena, Janet, Sarah, and the Gairdner Foundation for having me here in this incredible event. So my lab is interested in the neural mechanism of social and emotional behaviors. And today I would like to share with you two stories on social hierarchy and depression, which I think will make a good illustration on how optogenetics has enabled us to get to the heart of some of these complex problems. So, dominance hierarchy exists in almost all social animal species, ranging from fish to birds, from invertebrates to primates, even in kids as little as 2 years old when they form the playgroups. So for individuals, the social status has a great impact on health and one's quality of life. And social economic status has been identified to be the single strongest predictor of health. So in this famous Whitehall study conducted on more than 10,000 British civil servants, it was shown that the death rates as well as the disease rates for all kinds of diseases ranging from cardiovascular, diabetes, cancer, and to depression will gradually decrease as one moves from the lowest to the highest rank, even though as civil servants, they all have the equal access to the healthcare system. So the very first study conducted on social hierarchy was made by this Norwegian scientist called Abbey in the 1920s. When he was observing the relationship of the hens in his own farm, he noticed that there's a very strict pecking order such that only the high-ranked hens will peck the lower-ranked ones in a highly transitive and linear manner, and almost never the other way around. Abbey was also the first to propose that establishment of the dominance hierarchy can reduce unwanted fights, save energy, and promote social stability. So when we analyze this dominance behavior in the animal kingdoms as well as in human societies, we quickly realize that getting to the top of the hierarchy is not simply a matter of physical size or body size or physical strength or even aggressiveness. The personality traits, including courage, confidence, persistency, those sometimes referred to as grit, regulated by high cortical functions, also play an important role. So dominance can be displayed in a quite diplomatic manner, but sometimes this display can also go pathological, as shown in this epic handshake. So you see, this is actually a very typical gesture for the alpha male. And by the way, I heard the Prime Minister here dealt with this handshake pretty well. So operationally speaking, dominance can be defined by consistent winning at points of social conflict. So in order to create a situation of social conflict in a laboratory setting, we decided to adopt an assay called the tube test, in which we put 2 mice into a narrow plastic tube, and usually within 30 seconds, one of these mice will back out. So we will name this one as a loser and the remaining one as a winner. So for a cage of 4 mice, we can do 6 of these pairings, and based on the total number of winning, derive their rank order. So then, at that time, the question is whether such a simple assay actually reflects the dominance relationship of these mice. So several lines of evidence led us to believe that it does. So first of all, the tube test rank is highly transitive. If mouse A wins B and B wins C, then in 95% of matches between A and C, we found A would again be the winner. So this is pretty much like what Abby has described, this pecking order Abby has described for his chicken. And secondly, the rank is pretty stable. So here we measure the rank of the 4 cage mates repeatedly in 6 days. You can see there's some switch, but overall it is pretty stable. And finally, the tube test rank show a very good correlation with measures from several other dominance measures. For example, mice ranked higher in the tube test tend to consume more food in the visible bowel system, where food is relatively restricted. They tend to initiate more agonistic behavior in the new territories. Sometimes the rank 1 mice will trim the whisker of their cage mates. They tend to urine mark in more territories, and when seeing a female, they tend to produce more courtship ultrasound. So here's a typical ultrasound trace produced by a rank 1 versus a rank 4 animal when seeing the female. So having established tube tests as a simple and reliable measure for dominance, we were then able to ask, where is social hierarchy rooted in the brain, and which neural circuit can control the dominance behavior? So we had some clues from the human imaging studies, which showed that part of the prefrontal cortex is highlighted in the dominance-related behavior. So several years ago, my lab decided to use a series of molecular tools to tweak the synaptic strength of an equivalent brain area in mice called the dorsal medial prefrontal cortex, the DMPFC. And some of the molecular tools we used are the glutamate receptor, or the dominant negative form of the receptor. So with stereotypic injection using virus as a carrier, we can precisely express these molecules just in the MPFC area. So for example, overexpression of the R4 type of AMP receptor can greatly enhance the synaptic transmission, whereas the C term, the dominant negative form, can reduce the synaptic currents. Correspondingly, at the behavioral level, within 24 hours after viral injection, we saw that there was a corresponding increase or decrease in both the tube test and the ultrasound test, respectively, by these 2 constructs. So these results suggest MPFC-controlled temperament may contribute significantly to the dominance behavior. However, in order to show a direct function of MPFC, we still had 2 key questions remained. First is, how do MPFC neurons react in social confrontations in real time? And secondly, how quickly does MPFC activation change the behavior? the 24 time points is still too slow to distinguish whether MPFC controls the behavior by directly activating the relevant neural circuits or through a relatively slow hormonal control. So in order to address the first question, we set up the tetrode in vivo recording system in the tube test. So here each mouse is wearing a 16-unit tetrode, and the tetrode is pretty light, so the chronic implantation does not affect too much their daily life. And during recording, we further used a balloon to balance the weight of the recording cable, so the mice can perform the tube test almost freely without the extra weight on their head. So here I'm showing you one neuron recorded from this dominant mouse on the right. So you can see that right before this mouse started pushing, there was a strong increase in the firing of this neuron. So in more than 280 neurons we recorded, we saw about 10% of them showed a great increase during pushing or resistance, but not so much in retreat, suggesting that at least a portion of MPFC neurons are firing at the right time to potentially mediate this behavior. Now, for the second question, that is how quickly can the MPFC activation change the behavior, we turn to optogenetics. So here both mice are expressing this light-sensitive channel rhodopsin molecule in their head. So you can see that without light, they behave pretty normally. So the rank 1 mouse will quickly push rank 3 out of the tube. Now, in the next slide video, it's still the same pair of mice. The only difference is that we switch on the light for this rank 3 mouse as soon as it enters the tube. You can see the blue light shining, and with very little delay, in about several seconds, this mouse would start pushing quite fiercely, which is something it never did in previous trials until it pushed rank 1 all the way out. So rank 1 tried to push back once, but still eventually lost. So this happened in 5 out of 6 mice that we photostimulated. Some moved from rank 2 to rank 1. Some moved from rank 4 all the way to rank 1. And we noticed that for this rank 4 animal, we actually need to gradually crank up the laser power when it was competing against 3 and 2 and 1, demonstrating a nice dosage relationship between the level of MPFC activity and the amount of efforts required. So interestingly, although MPFC activation does not seem to change the muscle strength as measured by this grasp assay, it can turn a smaller mouse into a winner against an opponent that is 50% heavier, as you can see from their body size, illustrating a case where mental strength can overcome physical strength, if you would like. So that was the behavior on the photostimulation day. But when we continued the tube test, in the following days, we saw the mice behavior diverged. So about 2/3 of them maintained this newly acquired rank position, whereas other mice returned to their old status. When we compare the experience of these mice, we notice that those who have received more than 6 times of stimulated weaning all maintained their newly acquired position. But if their weaning time is less than 6, then some of them will return to the old rank. So this observation reminded us of this psychological phenomenon called the winner effect, where animals or individuals will increase their chance of winning after previous victory. So when Michael Tyson was released from jail in the 1990s, before he regained his championship title, his manager actually arranged for him 2 comeback fights, and each against a much weaker opponent to assure an easy winning. So the strategy his manager is using here is indeed based on the winner effect. We then did a series of experiments to show that this winner effect is mediated by medial dorsal thalamus input into the MPFC area. So with repeated winning, synapses in this neural pathway become strengthened in a long-lasting manner, also referred to as long-term potentiation. So far I've only talked about tube test, but in reality, animals are facing all kinds of competitions. So we then wonder whether the winner effect can be generalized or transferable from one behavior to another. So for that, we need to have a second dominance measure. And we were inspired by David Anderson when we were chatting about the behavior of his cats. He mentioned that his 2 cats will compete for this sunny, warm spot in his house. So inspired by this, a graduate student, Zheng Xiaofan, developed this warm spot competition essay where she put 4 mice on an icy cold floor, but with a heated corner. So here you can quantify the occupation time of the heat spot of the rank 1, 2, 3, 4 mice from the tube test. And you can see again there's a good correlation. The rank 1 mice from the tube test occupy most of the heat spot for most of the time. So with that, we were then able to do the transfer experiment where we can take the subordinate mice, which is inferior in both of these tests, subjected to 10 times of weaning in the tube test. Then we tested in this warm spot assay. So strikingly, even though this mice has never been photostimulated in this new context, its performance also became superior in this second test. So we believe that this kind of transferable or the generalization of the winner effect can help animal to reinforce their dominant status and help to set up the social hierarchy quicker. So before switching topic, I would like to acknowledge these 4 brave and super talented graduate students who have contributed to this social hierarchy work. Now for the second part, I would like to share with you our recent work on ketamine bursts and depression, which is related to how neuron and glial cell interact to give rise to this specific firing pattern to regulate mood and depression. So according to the World Health Organization, the prevalence of depression is surging over the years. So by 2017, it has surpassed heart disease and cancer, becoming the #1 most disabling disease in the world. Regarding the cause of depression, an old famous hypothesis points to a lack of monoamines in the brain, such as dopamine and serotonin. However, a major problem of this hypothesis is that although classical antidepressants such as SSI can very rapidly restore monoamine levels within hours, but the improvement of mood is much delayed, often taking weeks to months for the drugs to show efficacy. The situation may be changing with the emergence of this miracle drug called ketamine, which is hitting a lot of headlines and causing a big buzz in the pharma industry. So ingested to the classical antidepressants, ketamine can cause a very rapid antidepressant response, and in a large fraction, about 70% treatment-resistant patients. And the effect can kick on as early as 1 hour. So for this reason, ketamine has been argued to be the most significant discovery in the mental health in the last half century, because it may help reveal the core mechanism of this detrimental disease. So neuroscientists mostly know that a major molecular target of ketamine is NMDA receptor. But the conundrum is that NMDA receptor is expressed everywhere in the brain. So what exactly is the target, which brain region, and which cell groups? So towards this million-dollar question, people actually had a few clues. So first of all, ketamine is an open channel blocker, meaning that it can only block when the receptor channel is in an open state. Secondly, ketamine is also known to have a very fast metabolic turnover rate, so with a half-life of about 3 hours in blood. So this rapid hit-and-go kind of temporal profile suggests that ketamine is likely to act on a system that is intrinsically active and has NMDA receptors open. And thirdly, ketamine is also known to very rapidly increase the release of several neurotransmitters such as dopamine, glutamate, and serotonin, suggesting that this black box may normally inhibit these reward centers, which is the source of this neurotransmitter. So towards this idea, much attention has been focused on the GABAergic interneurons because these neurons are known to have a higher intrinsic firing rate. So in one prevalent model, it was proposed that ketamine blocks these presynaptic NMDA receptors of cortical interneurons, which then somehow lead to a cascade of changes, includes potentiated AMPA signaling, increased translation and relief of BDNF, as well as increased synaptogenesis. Recently, a metabolite of ketamine called HNK is also shown to be important for this action. So despite of some supporting evidence, however, if we look at the signaling processes, it is still quite complex and time-consuming, and it may not fully explain why ketamine can act so quickly. So in our recent work, we proposed an alternative model, a perhaps much simpler model, in which we suggest that ketamine blocks this NMDA receptor-dependent burst firing of an anti-reward center in the brain called lateral habenula, which normally inhibits the reward center through a GABAergic relay. And because this burst firing requires NMDA receptor, ketamine is going to silence the burst and take off this suppression brake onto the reward center. So the lateral habenula LHB has caused an exponentially growing amount of interest in the last 10 years due to this series of work showing that LHB encodes negative emotion, including stress, disappointment, and fear. And consistently, brain imaging studies showed that in depressed patients or in animal models of depression, habenula becomes hyperactive. So that's why sometimes LHB is also called the anti-reward center in the brain. So circuitry-wise, lateral habenula serves as a relay station that interconnects the limbic forebrain with the midbrain monoamine centers. Although majority of LHB neurons are excitatory glutamatergic, but through this GABAergic IMTG nucleus, as well as through feed-forward inhibition, it is known to inhibit the dopaminergic neurons in the VTA or the other serotonergic neurons in the dorsal raphe. Now, in the first part, I'd like to show you evidence on how ketamine regulates this burst firing to cause antidepressant. So we actually stumbled upon ketamine a couple years ago when a postdoc, Yang Yan, was trying this very difficult bilateral drug infusion experiment. Because there are some very dense fiber inside the habenula, so it actually took us some time and to make some technical adjustments in order to get this bilateral infusion with very high success rates. So at the time, Yang Yan was trying to test the antidepressant response of some antibodies in these congenitally learned helpless CLH rats. So these are rats that have been selectively bred by Fritz Hahn's lab for more than 20 generations to enrich the learned helpless phenotype. So every generation, he took the most depressed animal to mate, and then the offsprings are kind of born to be depressed. So because it was the first time we set up this challenging system, we wanted to include a positive technical control, and we reasoned at the time that, well, since LHB is hyperactive in depression, then maybe by blocking majority of its excitatory synaptic inputs with an AMPA receptor blocker, MBQX, we may cause antidepressant. But for curiosity, we also decided to try this NMDA receptor blocker, AP5, in a parallel experiment. So to our great surprise, AP5 caused a robust antidepressant response, whereas our originally intended positive control, MBQX, only caused a non-significant change. So we were surprised, but we were also confident with this result because we verified every single injection site with a CTB tracer. So knowing that ketamine is also a blocker for NMDA receptor, this serendipitous discovery led us to the research on ketamine. So here in both the false swing and the sucrose preference test, which model behavioral despair and anhedonia phenotype of depression, we found that ketamine causes a very rapid antidepressant response within 1 hour after infusion into the lateral habenula. It does not change the locomotion, and the effect can last for as long as 14 days. So to our knowledge, this may be the first demonstration that infusion of ketamine just into one brain area is sufficient to cause both rapid and sustained antidepressant response. So what does ketamine do to the LHB neurons? In a separate project, a very talented electrophysiologist, Yi Hui, was doing whole cell patch of these LHB neurons. So Yihui first confirmed that LHB neurons are intrinsically active. So on brain slices, without any external stimulation, this neuron can be categorized into 3 categories: the silent, tonic firing, and a small percentage of burst firing types. Yihui also confirmed that the bursting neurons tend to be more hyperpolarized than the other 2 types, which is an important point that I will return to later. Now, in the congenitally learned helpless rats, Yihui found the percentage of bursting neurons was greatly increased. So if she looks at the interspike interval, which is a reversal of the firing rate, she saw this new peak at 14 milliseconds, corresponding to about 70 hertz, which is caused by the burst. She also found in this second model of depression, the chronic restrained mice, almost exactly the same change. And notably, systemic injection of ketamine into this animal before the brain slicing can restore the bursting level back to the control. So at this point, this bursting result and the cannula experiment were still 2 separate projects due to a knowledge blind spot. But luckily, about 2 weeks later in the Cold Spring Harbor Conference, I heard a talk from Kuan-Hong Wang, who showed that NMDA can modulate burst firing of cortical neurons. So that was really a moment of enlightenment for us, because coming from the field of synaptic physiology, I tend to associate NMDA receptor with LTP and LTD. So I had overlooked the fact that this receptor is also important for burst firing in several brain areas. And indeed, due to its relatively slow decay kinetics, NMDA receptor can integrate synaptic inputs over space and time to support the calcium spike required for burst firing. We then decided to do this direct experiment by perfusing ketamine into neurons, into brain slices where we current clamp the neuron. So here I'm showing you all the spikes in the bursting mode in blue and all the spikes in the tonic firing mode in black. So you can see that essentially within 5 seconds after ketamine perfusion, this neuron was converted from burst firing to tonic firing. So the probability of bursting was reduced to 0. And we can completely mimic this effect by a more specific NMDA receptor blocker, AP5. And interestingly, consistent with the behavioral result, AMP receptor blocker and BQX only caused a minor, about 20% reduction in the burst firing, suggesting that bursting is more due to an intrinsic property of this neuron and only minorly modulated by synaptic inputs. Also, the classical antidepressant fluoxetine did not show any effect, at least within this time scale. In addition to NMDA receptor, we found burst firing also depends on a more hyperpolarized, a more negative membrane potential, as well as a low voltage sensitive T-type calcium channel. So one prominent feature of this T channel is that it has a very transient activation, but it can be deactivated once the membrane potential drops below minus 55 millivolt. And this special feature has allowed this channel to contribute to pacemaker activities in several rhythmic systems, including the heart to contract the muscle, or in the thalamus to regulate the sleep spindle. So we found this T channel is also abundantly expressed in the lateral habenula, and a block of TVSCC mibofraudial can also cause a major reduction in the burst firing. So in order to understand how T channel and NMDA receptor work together to regulate the LHB burst, we constructed this minimal biophysics model. So for the interest of time, I'm skipping some of these details. But essentially, but briefly, this is what we think is happening in the LHB neurons. So LHB, the membrane potential of these neurons are in generally more quite depolarized in the range of like between minus 50 and minus 60. So we think that when the membrane potential drops below minus 55 millivolt, it will help open the T calcium channel, and the initial calcium influx into the channel will help remove the magnesium blockade of NMDA receptor. As T channel quickly inactivates, NMDA receptor will take over the relay to further drive the membrane potential toward the threshold for the sodium spikes. And afterwards, as both channels inactivate and membrane potential drops below minus 55 millivolt again, it will prime the neuron for the next cycle of burst. So you may then wonder whether this LHB burst is sufficient to drive the depressive-like behavior. So for that, we again turn to optogenetics. So we first used a conventional method by giving high-frequency pulses of blue light. However, we found that LHB neurons can only follow the first of the 4 100 Hz pulses, probably due to some intrinsic limits of this, by physical limits of these neurons. Then, inspired by this rebound burst mechanism, we devised a strategy based on this hyperpolarization-based opsin, the MPHR. So by giving very short yellow light to transiently hyperpolarize the membrane potential, when the yellow light stops and the membrane potential rebounds, we were able to induce burst firing with about 90% success rate, as shown with both in vitro and in vivo optical recording. So using this yellow light protocol, we were then able to drive real-time place aversion and depressive-like behaviors. For example, when the yellow light was on, this animal will go directly into this floating, so sort of despair-like state. However, if we inject the mice with ketamine first, then they will keep trying and swimming because this rebound, MPHR-induced rebound burst, also depends on the NMD receptor. So far I have told you how ketamine blocks LHB burst to cause antidepressant. But what is responsible for the increase of LHB bursting in depression at the first place? So it turns out the answer may come from a proteomics screen we performed almost 10 years ago. When I first set up my lab, we decided to collaborate with John Yates' lab at the Scripps Institute to look for proteins that show differential expression in the habenula of CLH rats. So we identified about a dozen proteins, and in 2013, we published one of this molecule called beta-CAMK2. But among the upregulated protein, there's this other interesting potassium channel, which is astrocyte specific, which is a channel that is specifically expressed on astrocytes. So again, for the interest of time, I'm skipping a lot of Western verification and functional experiment, but just want to bring your attention to this striking cellular phenomenon in which when we just overexpress this KER channel just in the astrocytes of LHB, we were able to drive hyperpolarization and burst firing in the neuron. So although today I'm telling you the story in this order, but in reality we sort of discovered this striking phenomenon first, then we got into burst firing and ketamine. So then the question is, how on earth can a channel on astrocyte regulate the membrane potential and the firing pattern of the neuron? When we looked at the expression of KER channel, we were puzzled for a while because it seems to co-localize much better with the neuronal marker nu-n than with the astroglial marker. Even though in the same brain slice, just in the neighboring hippocampal area, we get very nice co-localization with the glial marker. When we zoomed into the LHB neurons, we found this green KER signal tend to wrap around the nu-n signal. With an even higher magnification EM, we saw that these KER-positive gold particles were evenly distributed on the membrane processes that enveloping the neuronal soma. Finally, with cell type specific knockdown, we prove that KER is indeed expressed on astrocytes, not on the surface of the neurons, because only when we knock it out on astrocytes, the signal disappeared. So pretty much like what Raymond Cahal painted in this amazing picture almost 100 years ago, in lateral habenula we found astrocytes are wrapping around the neuronal soma. And within this highly confined extracellular space, there is this potassium channel that is actively clearing the potassium that is released from the bursting or the firing neurons. And we predict that if we lowering the capacitance of this clearance, we should reduce extracellular potassium or the K out. Then based on the Nernst equation, we should cause an increase, a depolarization of the membrane potential. And that is indeed what we found when we use barium to block KER channel. Within minutes, we can cause a membrane depolarization. We can also do the conversely by using TTX to block the neurons from firing. We can decrease extracellular potassium and hyperpolarize the membrane potential. And interestingly, the amount of change in the membrane potential caused by this drug positively correlates with the initial firing rate of the neuron. So sort of the more active the neurons are, the larger the contribution from this potassium, the KER-based potassium buffering mechanism. So to quickly summarize this take-home message, we think that in highly active neurons, such as LHB neurons, a very efficient potassium buffering mechanism is in need as a neuron at its extracellular junction. With prolonged emotional stress, the upregulation of KER4.1 will decrease extracellular potassium and hyperpolarize the neuron. Then due to the synergistic interaction between the T calcium channel and NMD receptor, it will convert tonic firing into burst firing. And because we know that bursting is more efficient in information transmission because it can reduce synaptic failure or synchronize the neural network, it will then lead to a stronger suppression output onto the reward center to cause depression. And finally, ketamine, by blocking this NMD receptor, can reverse bursting back to tonic firing or even silent state, therefore taking off this suppression brake onto the reward center to combat depression. So one strong prediction from this model is that drugs against the T channel should also block bursting and cause antidepressant effect. And that's indeed what we found with these two different drugs, two different inhibitors of T channel, one systemically injected and the other locally infused. And indeed, one of these drugs is called esosaximide. So it is still used in clinic to treat absence seizure in the kids. So we are now just initiating a small clinical trials with our affiliated hospital to test this drug on depressed patients. So with that, I would like to thank the people who did this work and our collaborators, without whom all this work would not have been possible. Thank you very much.
Thank you, Dr. Hu. That was a fascinating talk. If only it were as easy as shining a light on Donald Trump's head to modulate his social dominance. We have one more speaker before we take a quick break. So please save up your questions because there will be an opportunity within the break to interact with the speakers. Our next speaker is Dr. Yang Dan. Dr. Dan is a Paul Lick Distinguished Professor in the Department of Molecular and Cell Biology at the University of California, Berkeley, and a Howard Hughes Medical Institute investigator. She studied physics as an undergraduate student at Peking University and received her PhD training in biological sciences at Columbia University, where she worked on cellular mechanisms of neurotransmitter secretion and synaptic plasticity. Dr. Dan did her postdoctoral research on information coding in the visual system at Rockefeller University and Harvard Medical School. Dr. Dan's recent interest is understanding the neural circuits controlling sleep, a fundamental biological process with a strong impact on human health. Please welcome Dr. Dan to the stage.
First, I'd like to thank the organizers for giving me the opportunity to present at this great event. So I'd like to talk about the neural circuits controlling sleep. Sleep is an essential innate behavior. It's innate because we know how to do it when we're born. No learning is required. In fact, this baby here probably sleeps better than most of us in this audience. It's essential because, as far as we know, all animals sleep, and that even includes worms and flies and even jellyfish. And not sleeping enough causes cognitive and emotional impairment, and also many other health problems, including obesity and cardiovascular diseases. So, mammals, birds, and reptiles have 3 distinct brain states, in fact, 2 types of sleep, and both can be distinguished from wakefulness based on just EEG and EMG recordings. Is there a laser pointer somewhere here? Oh, it's the mouse, okay. So during wakefulness, the EEG is in this what they call desynchronized state, meaning that it's dominated by low amplitude, high frequency activity, and the EMG shows a high muscle tone. During non-REM sleep, which is also called slow wave sleep, the EEG is dominated by these large amplitude slow waves, and the muscle tone is reduced. During rapid eye movement or REM sleep, the EEG is again desynchronized. It's actually quite similar to the wakeful EEG, but there's a complete skeletal muscle paralysis. So the idea is that this desynchronized EEG allows us to experience vivid dreams, but the REM paralysis can prevent us from acting out our dreams. So the normal sequence is that we go from wakefulness. First, we fall into non-REM sleep, and then we alternate between REM and non-REM sleep, about 4 or 5 cycles per night. So we're interested in both REM and non-REM sleep, but today I'm going to focus on this first step, which is to fall asleep from wakefulness. So what controls sleep and wakefulness? Pioneered by the work of Marusi and Magan, who discovered the ascending arousal system, we now know that there are multiple neuromodulatory systems that promote wakefulness. So these include the cholinergic system in both the brainstem and basal forebrain, multiple monoaminergic systems such as dopamine, as we heard, noradrenaline, serotonin, and histamine. There are also neuropeptide systems. The better known one is orexin or hypocretin. And if we lose that system, we have narcolepsy. And in these subcortical regions, including the brainstem and hypothalamus and basal forebrain, there are also many glutamatergic and GABAergic neurons that promote wakefulness. In fact, this was a shock when we first got into the sleep field 8 years ago. It's actually very easy to find neurons in the brain that promote wakefulness. If you do optogenetic experiments, you can almost go into these brain regions and pick a random population of neurons to activate, and chances are the mouse will wake up. In contrast, we know much less about the sleep circuit. So this is a review written 13 years ago, and they highlighted a single region called the preoptic area in the anterior hypothalamus, and I'll call it the POA. So we know that a subset of the neurons in the POA, so these are GABAergic neurons, are preferentially active during sleep, and their activity promotes sleep probably by inhibiting a lot of these well-known wake-promoting neural populations. So the main interest of my lab is to figure out what controls sleep. To me, this is a pretty straightforward problem. All we need to do are just 2 things. One is to find the sleep neurons, and then we have to map their connections. And today I'm going to focus on the first step, which is to find the sleep neurons. Because to me, once you figure out who these neurons are, it's pretty straightforward to map their connections. You can also, do a lot of things to manipulate the activity of the sleep neurons to enhance sleep. So we have 2 basic criteria for sleep neurons. One, they should be sleep active, meaning that they should be more active, fire more action potentials during sleep than wakefulness. 2, they should be sleep promoting, meaning that their activation should increase sleep and their inactivation should decrease sleep. So in the last few years, we have made a lot of progress to figure out who the sleep neurons are in the POA. But we and others have also found that there are sleep neurons outside of the POA. And that suggests that perhaps the sleep control network is a lot more distributed than we originally thought, as suggested by the simple model with a single sleep center. So a couple years ago, we decided to perform a systematic whole brain screening for sleep neurons. So there are two strategies of doing this. One is that we can screen for sleep active neurons and then see whether they're also sleep promoting, or we can screen for sleep promoting neurons and then see if they're sleep active. So we have taken both approaches, and I'll give you an example of each approach today. So to screen for sleep active neurons, we use this technique developed by Li-Chun Luo's lab. It's called a false trap. So we crossed two mouse lines together. One of them expresses Cre-ER under the false promoter. So we know that when the neurons are activated, they express this immediate early gene false. So in this mouse line, those active neurons should express Cre-ER. The second mouse line expresses a fluorescent marker, GAP in this case, and in a Cre-dependent manner. So we divided these cross mice into two groups. One group is called the SD mice, so they're sleep deprived. So the idea is that for this wake active neurons that are active during sleep deprivation, right? So we keep them awake. The injection of tamoxifen causes the Cre-ER expressed in these wake active neurons to enter the nucleus, and that causes a permanent labeling of the wake active neurons by GAP. In the second group, we call them the RS mice. So these are recovery sleep mice. We first deprive them of sleep for 6 hours to make them really sleepy. And then we paired injection of tamoxifen with recovery sleep to label the sleep active neurons permanently. And when we compare these two groups of mice, we saw many brain regions with differential expression of GAP, which is hardly surprising. So these are the different regions. So some of them express more GAP in the RS group, and others express more in the SD group. Now, when we looked at our top candidate, so this is the region with the highest relative expression of GAP in the RS group. This is the region shown here. So this region with this shape of an inverted pear is called the PAG, periaqueductal gray. So this little hole here is the aqueduct with the cerebral spinal fluid. So you can see that in the ventral PAG, we see a lot more GAP neurons in the RS group than the SD group. And when we saw this effect, we actually got quite excited. And that's because in the sleep field, everybody knows about von Economo, who was the first one to propose the sleep center in the POA, which I mentioned. Everybody knows about the POA. But in fact, several decades before von Economo, this guy here, Ludwig Mausner, so he's also the first person who described the Mausner cells in the fish. So he actually proposed a sleep regulating center near the oculomotor nucleus, which is exactly here. So the pair of oculomotor nuclei are just about here and here. So the sleep neurons that we found are exactly in the region that Mausner proposed. Now, it turns out that when Mausner died, just a few years after he proposed the sleep center, the idea of a sleep center remained very influential. In fact, von Economo was very much inspired by Mausner's idea of a sleep center, and that's why he went and looked for his own sleep center. But the particular center proposed by Mausner was largely forgotten, so he was a little bit ahead of his time. So when we found these putative sleep neurons in this region, we got quite excited. Now, obviously, even though there are a lot more neurons labeled in the RS group than the SD group, these are still a very small minority of all the neurons in the ventral PAG. So we have to figure out who they are. So for that, we did gene profiling, which Leslie also talked about. So in this case, the mouse line that we used expresses not only GAP, but also a large ribosomal protein called L10A. So if we use an antibody against GAP, we can pull down L10A together with the mRNA attached to it, so we can do the sequencing. And this is what the data look like. This horizontal axis is the relative expression level of the gene, right? So each dot is a gene in this putative population of sleep neurons relative to the entire chunk of tissue that we cut out. And then the vertical axis is the significance level. So we see a number of highly enriched genes in this red box here. And we decided to focus on neuropeptides. And there's 3 of them in this box. One is called CALCA, so this is a calcitonin gene-related peptide. It's also called CGRP alpha. There's another one, CGRP beta, and the third one is galanine. So when we looked at expression of these 3 peptide markers, we saw that basically CGRP alpha and beta overlap by more than 80%. So these 2 peptides, which are highly related to each other, also pretty much label the same population of neurons. Galanine is expressed in a much smaller population, and more than half of them also express CALCA. So we decided to focus on CALCA as our putative marker. And when we express the expression of CALCA with a GFP labeling of the putative sleep neurons, we saw that about 70% of them express CALCA. So it seems like CALCA is a good marker for these putative sleep neurons. So the next question is, are the CALCA neurons really sleep active? So even though they are labeled by CFOS, but we know that CFOS depends on many other factors, so it's not just activity. So for that, we did recording. And to make sure that we're really recording from a neuron that expresses CALCA, we use optogenetic tagging. So in this case, we use a CALCA Cree mice. So these are generated by Richard Palmiter's lab. And we injected channelodopsin with Cree-dependent expression of channelodopsin into this mouse so that we can label the CALCA neurons. And then for recording, we use something called the optrode, which consists of an optic fiber surrounded by several bundles of electrodes. So here you can see one really nice unit. So each line is an action potential, and each little blue dot is a 5 millisecond laser. So the fact that this neuron is reliably driven by the laser suggests to us that this is a neuron that expresses channelodopsin, therefore it should be a CALCA neuron. And once we find a neuron like this, which by the way is very difficult, so on average we probably find 1 neuron per, every 3 days, sometimes a week. But once we find it, we turn off the laser and just record its natural firing rate. So you can see that here is one recording. So this is the power spectrum of the EEG, and this is EMG. And so based on these 2 recordings, we can classify the brain stage, which is color-coded here. So this is all done automatically by the computer. And this is the firing rate of this cell. So you can see that this neuron has a high firing rate during non-REM, right, orange, but the firing rate is low during both wake, gray, and REM, which is blue. And here's a summary of 34 cells, I believe. So each line is 1 cell across the 3 brain states. We also summarize these data in this 2-dimensional plot. So the horizontal axis is the firing rate difference between non-REM sleep and wakefulness normalized by their sum. And the vertical is the rate difference between REM and wake. So the identified units, so these are the CALCA units, the blue dots, are mostly on the right-hand side, indicating that they're more active during non-REM sleep than wakefulness. But the unidentified, these gray units, which are recorded in the same brain region, but not driven by laser, so we don't know what cell type they are. And you can see that they're much more scattered. And if you want to contrast these 2 populations, focus on this histogram on the top. So you can see that the blue units are really peaked on the right-hand side. So these are non-REM active, but the gray units are really much more distributed with a lot more wake-active neurons. So basically, these experiments told us that the great majority of the CALCA neurons are indeed non-REM active. So this is our first criterion, they're sleep active. Now the question is, are they sleep promoting? So this is an experiment with optogenetic activation, channelodopsin. So this is, again, the EEG power spectrum, EMG brain state. So we turn on the laser for 2 minutes per trial, 10 hertz stimulation, these blue shading here. And this is just one raw experiment, and this is a summary of all the mice that we tested with all the trials. So each line is the probability of each of the 3 brain states before, during, and after laser stimulation. So you can see that during laser, right, this is just 2 minutes, there's an immediate increase in non-REM sleep and decrease in both wakefulness and REM sleep. And then as soon as you turn off the laser, the brain state goes back to the baseline. We also did inactivation. So in our hands, IC++, which is a mutated channel rhodopsin responding to also blue light, but it's a chloride channel, works the best. So when we inactivated these neurons, we saw decrease in sleep and increase in wakefulness. And in addition to this kind of acute optogenetic, very quick activation and inactivation of these neurons, we also tested the effect of slower activation and inactivation using chemogenetics. So these are dread experiments. So this is the excitatory dread. The open circles are the control experiments. And you can see that if we activate these neurons, right, over a time course of a few hours, we saw increase in non-REM sleep during the first 2 hours, decrease in wake, and not much change in REM sleep. And if we inactivated these using the inhibitory dread, we saw the opposite effect. So basically, we have optogenetic and chemogenetic activation and inactivation. And all of these experiments indicate that the cochlear neurons are non-REM promoting. So this is our second criterion. So far, we've been using CALCA, which is a neuropeptide, as a marker. But it turns out that in these brain regions, most of the neurons expressing these peptides also use traditional neurotransmitters. And it turns out that the CALCA neurons, about 60% of them are glutamatergic because they express this glutamatergic marker VGLU2. But the other 40% are actually cholinergic neurons. So for the sake of time, I won't show you the data, but basically we figured out that the sleep effect of the CALCA neurons are mostly caused by the glutamatergic subset, not the cholinergic neurons. So this is also very exciting to us because so far, most of the published sleep neurons that we know of are actually GABAergic, which is also why most of the sleeping pills are targeting the GABAergic system. So finding these glutamatergic excitatory neurons that promote sleep is really quite novel and exciting to us. But it turns out that the CALCA neurons are not the only sleep-promoting glutamatergic neurons in this region. When we looked at another marker, CCK, which is another neuropeptide, so we already know that the CCK is a great marker for sleep neurons in the POA. And it turns out that CCK is also expressed in the ventral pack. They are about 100% glutamatergic because more than 95% express VGLU2. But they're entirely separate from the CALCA neurons. So here, CALCA is red and CCK is green. So they're sitting right next to each other, but they're totally non-overlapped. And when we looked at the CCK neurons, also activation caused increase in non-REM and inactivation caused decrease in non-REM. So the CCK neurons are also glutamatergic, they also promote sleep. And this is not all of the glutamatergic sleep neurons yet. So just a few hundred microns behind or posterior to the CCK and CALCA neurons, we found another marker, which is a neurotensin, NTS. So this is just data from the Allen Institute, and you can see that these clusters of NTS neurons are also in the ventral region of the pack. And when we tested the NTS neurons, activation also increased non-REM sleep at expense of both wake and REM, and inactivation caused the opposite effect. So just so the NTS neurons are non-REM promoting, but are they non-REM active? And in addition to the optical recording experiments, which are extremely low throughput, we also use optical imaging. So in this case, it's GChem imaging that Leslie also talked about. But because these neurons in the pack are very deep in the brain, we have to image through this green lens or microendoscope. And we use this miniaturized microscope developed by Mark Schisser's lab to image this activity. So you can see that instead of optical, you see one neuron at a time, we can actually image activity of a bunch of neurons at the same time. And these are the calcium traces. So you can see that these NTS neurons are more active every time we have a non-REM sleep, but they're shut down during REM and wakefulness. So just to quickly sum up what I showed you so far, in the effort to screen for sleep-active neurons, we have so far found 3 populations of glutamatergic non-REM neurons, CCK, CALCA, and NTS. So I don't have time to show you the data, but we think that they're actually reciprocally connected to each other, and all three of them actually project to the POA, which in turn inhibit the wake-promoting centers. So we know that the POA sleep neurons are GABAergic. So we think that perhaps, after all, Mausner was right, that the ventral region of the PAC is indeed a sleep center. It's just a excitatory sleep center that's upstream of the POA, which is the more famous sleep center proposed by von Economo. So this is our effort to screen for the sleep-active neurons. And now I'm going to switch and talk about this other technique that we use, which is to screen for the sleep-promoting neurons. So our idea is actually very simple and straightforward. We're going to start with the wake-promoting neurons. As I said earlier, we know quite a few of them. And we're going to use retrograde tracing to find the neurons that are upstream of these wake neurons that inhibit them. Because if you have a neuron that inhibits wake-promoting neurons, then chances are your activity can promote sleep. And once we find these inhibitory sleep neurons, we're going to trace one more step back and look for their excitatory input. Because if you can actually excite other sleep-promoting neurons, then you could be promoting sleep yourself. So it's a very simple logic. So this was pioneered by Chen-Yan Ma, a postdoc in my lab. First, what Chen-Yan did was to target a whole bunch of wake-promoting neurons and look for their GABAergic input. In particular, the GABAergic neurons that broadly inhibit many wake-promoting neurons are considered strong candidates for sleep neurons. So in the initial screening, Chen-Yan targeted 7 populations of wake-promoting neurons, including histaminergic, noradrenergic, glutamatergic, GABAergic, and cholinergic neurons in a whole bunch of brain regions. And she found a whole bunch of regions with broad GABAergic inhibition to these wake-promoting populations, including the POA, right? So this actually makes sense. It's just sort of a sanity check. And in addition, Chen-Yan also found this region, which is a central nucleus of the amygdala, which came as a big surprise because the CEA has been studied in terms of fear conditioning and fear learning. But it turns out that CEA also contains a population of neurons that promote sleep. So I mentioned just a few slides ago that there are these NTS neurons in the PAC that promote non-REM sleep. But in fact, Chen-Yan was the first person to identify NTS as a marker for the CEA sleep neurons. And now we actually know that NTS is a general marker that marks at least 5 or 6 different populations of non-REM neurons. But for the sake of time, I'm not going to show you the data that Chen-Yan collected, which is really a beautiful set of experiments. Instead, I want to tell you about this third candidate, which is the substantia nigra pars reticulata, or SNR. So this structure was studied by Dentian Liu, another brilliant postdoc in my lab. So Dentian first did optogenetic activation and inactivation. So here's the effect of activation, and you can see that there's increase in both non-REM and REM sleep and decrease in wakefulness. So the SNR neurons are a little bit different from the PAC neurons that I told you about, which only promote non-REM sleep. And here is optogenetic inactivation, huge increase in wakefulness, right? So almost 100% completely suppressed sleep. We also did a chemogenetic activation, increase in both non-REM and REM, and inactivation, decrease in both. So again, with the bidirectional manipulation, we know that these SNR neurons promote sleep. Now, so we were led to the SNR based on this anatomical screening to study their sleep effect. But in fact, the SNR has been studied a lot, but mostly in terms of controlling movement. So how can the same population of neurons control both sleep and movement? And to get an idea of how that works, we analyze the natural motor behavior of these mice in their home cage. So based on video recording and also EEG and EMG recordings, we broadly classify their behavior into 4 states. Locomotion, LM, which is when they're running around, easy to tell. Non-locomotor movement, MV, which is basically eating, grooming, or some posture adjustment. quiet wakefulness, so they're not moving, but based on EEG, we know that they're not asleep either. And finally, sleep. And when we looked at the transitions between these behaviors, we realized that they're highly non-random. So for example, we never see that the mouse is sort of running around and bam, they fall asleep, right? That just never happens, right? Instead, if they start out running, they always transition into non-locomotor behavior, just sort of like stop moving, stop running, but then sort of local movement. And then from a movement, they either go back to running or they go into quiet wakefulness. They still don't go to sleep right away. So in other words, if you rank these behavior states in the order of sort of decreasing movement and also arousal, then most of the transitions are between neighboring states, right? So they either stay in the same state or they move to the next state, right? Either up or down this chain. The only exception is sleep to movement. So sometimes they skip the quiet wakeful state. They wake up, they start moving. And when we looked at the effect of optogenetic activation, we saw decrease in locomotion, also movement, and increase in sleep, as we showed before. But this is not due to the direct transition from these movement states into sleep directly. In fact, we never see a transition during optogenetic activation that we normally don't see during a natural state. Instead, what happens with optogenetics is that we're facilitating the transitions down the chain, right? So from locomotion to movement to quiet wakefulness to sleep, and never sort of a jump that we normally don't observe. And when we inactivated these neurons, we basically push the behavior state in the opposite direction. So in other words, the activation of these neurons is not causing these weird transitions, right, unnatural. Instead, we're just biasing the normal transition probability. So this gives us the idea of how the same population of neurons can both control movement and sleep, because you can almost rank these behaviors in the sort of the same chain. Okay, so now we have found one population of neurons that control sleep, and as promised, we're going to trace one more step back and look for their excitatory input. So Dan Qian targeted these SNR GABAergic neurons and looked for their excitatory input. And one population of excitatory input that we see is actually hardly news for the people studying the SNR, which is the subthalamic nucleus, the STN. So in the basal ganglia field, actually everybody knows that the STN is a major input to the SNR, except that we didn't study that circuit, so we have to do our own tracing to find out. Now, the interesting thing is that the STN neurons, so these are glutamatergic, but they also express the neuropeptide NTS, which I mentioned before, both in the PAG and also in the CEA. And when we activated the STN, it's getting confusing. So STN stands for subthalamic nucleus, and NTS stands for neurotensin. So when we activated these neurotensin neurons in the STN, we also saw increase in non-REM sleep, and we can also activate their axon terminals projecting to the SNR, right? Because these neurons are upstream of the SNR, we saw the same effect. Okay, so just to sum up this part of the talk, using this systematic screening, right, two-step screening, we first found the SNR GABAergic neurons, which innervate multiple wake-promoting populations. So these neurons promote sleep. And then tracing one more step back to look for their excitatory input, we found the subthalamic NTS neurons that also promote sleep. So I have to say that my lab started working on sleep about 8 years ago. And the first 5 years was incredibly frustrating because we studied sleep, but we were just not finding any sleep neurons, just because the sleep neurons are usually outnumbered by the wake neurons. And only in the last 3 years or so, using the two types of screening, right, both for sleep active neurons and sleep promoting neurons, we were finding these sleep neurons just one after another. So it's really, the effort really paid off, and it's quite exciting to us. But it also begs the question of why so many populations? Why do you need so many of them? Why not just a single sleep center? Is there any logic to this? We actually think that there's a very simple logic to this. So we think that the fundamental purpose of sleep is to shut down movement, and we call this the motor theory of sleep control. Now, if you think about it, the first sign that somebody's asleep is that they're not moving. And this kind of explicit physical movement is controlled by the somatic motor system, and we call that the action system. Now, there's another type of movement, harder to see, but equally important. So they include things like breathing, heartbeat. They're controlled by the autonomic motor system, and we call that the arousal system. So we call this the arousal action, the AA model. Now, when we fall asleep from wakefulness, the autonomic motor activity slows down quite a bit, and the somatic motor system basically shuts down. And when we look at the circuit, right, after all the screening effort, all these multiple population of sleep neurons, most of them are actually associated with these two motor systems. For example, the STN, subthalamic nucleus, and SNR, they're part of the basal ganglia motor circuit, right? They control somatic movement. And the PAG, which I talked about in the first part, is a very important node of the central autonomic network. So this is just a diagram from the Kendall book. Now, although I didn't have time to show you the data, we also found sleep neurons in the hypothalamus, amygdala, which I mentioned very quickly, and also the parabrachial nuclei. So these are all major nodes in the autonomic network. So we think that the reason why the sleep network is so distributed is because these two motor systems are very distributed. So in order to control them effectively, the sleep control mechanism basically infiltrates and becomes part of these motor systems. So this idea also has direct implications for why we sleep at all, right? So what's the function of sleep? We think that the function of sleep is to promote metabolic processes that are fundamentally incompatible with motor activity, which is why sleep has to shut it down. So we think that it has to do with repair and rejuvenation of cells, both inside the brain and in the rest of the body. Now, of course, even with the simple purpose of shutting down movement, the network is still very complicated with multiple brain regions and multiple neuropeptides. But we think that they're not all created equal. In terms of the brain regions, we think that the PAG, the ventral region of the PAG, is a particularly important region coordinating the entire sleep control network because it has multiple glutamatergic neurons that send these long-range projections to other sleep-promoting regions. And in terms of neuropeptides, we think that the NTS is a particularly general marker that marks multiple populations of sleep neurons, spanning both the arousal and action systems. And finally, these are the people in my lab who actually did the work. So Joy is the one who did the screening for sleep-active neurons, Tianyan screened for sleep-promoting neurons, and Dan Qian is the one who studied SNR, STN. These are the early pioneers in my lab who got all the techniques to work, and all of them are running their own labs right now. We also have multiple collaborators, in particular Ed and Carl, who are being honored at this event. I think it's fair to say that without optogenetics, I wouldn't be studying sleep at all. So this is really a technique that completely redefined my career. And thank you for your attention.
Thank you, Dr. Dan, for a wonderful talk. We're going to take a quick break right now, so feel free to grab a coffee and stretch your legs. We'll meet back here at 11:15 when we'll start up again. Please use this opportunity to ask questions. We'll do it very informally, so if you want to come to the front of the stage, the speakers are happy to answer any questions you have.
Try one more time. See how it looks like.
Do you have an HDMI on here? No, we don't.
Thank you.